Hi, I'm going to explain RHO gene location and its mutation in this video. Although every cell in human bodies has RHO gene, it's turned on mostly in rod cells in retina of eyes to produce rhodopsin protein. I'll give you more information about rod cells and retina in the function part on page 2. But for here, RHO locates on the longer arm here of chromosome 3, band 2, subband 2, and subband 1, as I labeled in this one in purple. You might feel confused about what are these bands in different levels. On the microscopes with different resolution, artificially stained bands can be observed. Application of higher resolution allows observation of more precise bands. So for example, subband observation needs higher resolution than subbands. RHO gene composed of 6,706 DNA bases that are nucleotides, labeled by letters G, T, A, and C. The most common and first determined mutation of RHO gene is C68C substituted by A, as I labeled in yellow here, which means a substitution of base C into A at the 68th position in the coding DNA. I mentioned coding here because there are actually non-coding regions in DNA. In RHO gene, there are 5 exons and 4 introns. Exons are coding regions with meaning. So as you can see, I labeled in purple here. There are 5 bands in purple, and exons and introns are meaningless region between 2 exons. So the bands in white, 1, 2, 3, and 4. Which will be removed during transcription. So genes aren't directly changed into proteins. It's firstly converted into mRNA via a process called transcription that takes place in nucleus in human cells. The mRNA will then be transported out of the nucleus into the cytosol to produce rhodopsin protein via translation of the code brought by mRNA. In general, 3 neighboring bases are bundled together as a group called codon, here codons, that will finally be transferred into one protein. Let's come back to this figure. The C68 C substituted by A mutation on RHO gene changes its codon composition. As a result, the original protein proline encoded by CCC, proline encoded by CCC, is changed into histidine encoded by CAC, which occurs at the 23rd position on rhodopsin protein. Thus, the protein mutation is called P23H. I'll introduce more details about rhodopsin protein in the following pages. See you there.